的时候的，出
不是
差越
你哪天
瘦越快。
回来的呀？
嗯，
你哪天回来的？
周六一个星期。
你去了礼拜是吧？
对，连来加去其实一个星期，因为他那个会是3天，然后又加了2天在他们那个 institute，等于是
你没到别的地方去转转？
没有太那个。我这不是还回来忙这个？
你有啊？哟，这赖我身上是吧？你可以在那儿也可以啊。你看小玉上次不就是在澳大利亚，我这不是给他主持的吗？对吧？你
哦
这不是。
哦哦，倒也
对啊，你这
是。
不是借口啊，这是他上次人在那儿，我给他主持的呀。
我应该这么说，我应该那啥。
哎呀，反正那怎么样啊？那边儿
挺好的。他们的那个代谢的那个 facility 其实弄得非常好，就
那
是他们整个的就是 core facility 跟动物相关系的，尤其是动物生理、动物的，就是还有 mass spec 的 core，还有那个
你是去的哪个地方？我都没问
monash。
啊。Mona，
是他会议是那个 long 是那个 long 的那个会议的 long conference 的系列，就是 metabolism 那个系列。然后又去了 Monash，他们那个 BDI，就是那个把什么，他叫 BDI 叫全称叫什么来着？
人家叫你去开会，你连人家名字都叫不出来。
这记得住。我是那啥，这 seminar 之前有点激动，经常那什么，反正就是把 medical medicine BDI，
啊。
就是
那
就是很多代谢的
那
那个 cluster 还是非常好。
他们有啥谁做的不错的吗？
他们做代谢有挺多人的，那会上其实不光是他们 institute，那他会上其实是一个 international 的会，包括澳大利亚的，包括那个悉尼的，那个墨尔本的，然后还有北欧的，美国的也有，那个美国有几个来的，就我以前的 colleague 也有，那个 Paul Cohen 还有 Zack Knight 都去了，然后还有那个 NIH 做 microbiota 那个女孩，
哎，
那个做 microbiota 的那个女生，
男的女的，
女生
老的
做
年轻的，
年轻的。
年轻的，
嗯
年轻的有谁做？
HHMI 一个 HHMI。
不是 NH 哪有 HHMI 啊？
不是，哎，不对，哎，他是以前 Janela Farm 吧？他以前在。
你也
等一下，我看看。
你这都记串了。
嗯，
这
是不是叫那个叫什么蒙的曾老师？不是，是个美国人。
哦，美国人啊。哦，OK，
嗯，
郑老师早，不好意思，刚刚我就不打断你们这 conversation。
哎，你早，我们正在调机器，没事。
哇塞，
哎，
不是你要不要试一下你的 PPT？
对，我正在说呢，正在想说我的 PPT。
你套个可以录像对吧？我跟你说一下啊。
啊，
不可以
可以，
可以是吧？可以啊。
哦，都没问题。
OK，
但是那个，哦，那对。
但是怎么了？
录像的话，我得经常想一下下一句是不是有什么词儿不该说之
哎呀，
类的。
没事儿，这没你这你
那我看，
你又不是上台领奖，这没关系，这随便说，我们都很 casual 的，这个都是非常随意的。不，你认识李豪吗？
认识，
啊，OK。
我怎么见过？
我们好像没有见过这个本人啊，曾老师。
哎，
我们应该
上次在上海，你没去，之前有个代谢的会。哦，
symposium 嘛。
你没去，但是我怎么感觉我就是 online interaction 有好多次，对吧？我的 PPT 可以看到，对吧？
可以，没问题。
行行行。
不，你
嗯，
你一个人都想不出来吗？这个跑了一圈儿。
那个主要是它这个 focus 也不一样，它主要还是做 就纯的 metabolism 或者 neural metabolism，就是跟那个就比如说做 brain 的不同的 region 啊，做那个 hypothalamus 啊，做这些特别多。再有就是特别 metabolism，就是比如说做那个不同 organ 的 metabolism，
OK，好吧，
其实可能
那
也能够 match 起来，就是有几个偏向于做 brain body interaction 的，可能也是有这方面，也能够 related。
好吧，那我
嗯
回头再详细问你呗。
嗯，
今年还出差吗？
今年都是国内的了。
还有1个月。
对呀，哎呀，我看看，我我将来2个星期大概还有100张 PPT 要做，而且都是没
我的个
做。
妈呀！哎，你太忙了。
我觉得大家都很忙，那个 Paul 是不是也很忙？就是
应该
要
到年底应该都很忙吧？就是各种的这种述职
嗯，
报告、年底会议、交差解体啊。
对，中期汇报、年度汇报，年度述职、年度总结，就是都
哎呀，
都在都这样。
可能美国现在是 Thanksgiving 对吧？就是非
很明显，
常的 relax，
美国，
国内反过来。
美国，我们单位都快解散了，这当然 relax。
没有没有，开
对，
玩笑的，不会的。
那是。
哎，你说他要是越那啥，你们是不是越就是对科学的热爱会更加强烈啊？就终于可以就比较
你说你说没有压迫就没有反抗吗？
不能这么讲，就是说就是你内心的感觉会表达的更强烈，
我你你你想
就是
让狗
可以特别敞亮的热爱科学的那种感觉。
还特别是啊，屋顶都快没了，还当然敞亮了，怎么能不敞亮呢？
因
我们
为到
这个，哎
到
呀，
对啊，危急的时刻不是真爱，对吧？就很难就那啥，
我觉得你现在有点儿那个啥，
嗯，
站着说话不腰疼感觉。
没有这个要涅槃，跟你说。
你别看我们这些在美国的同学水深火热的，
涅槃之后变凤凰，我们
哎呀，
都是哎，
行行行，你说先烧成灰是吧？
这才能变凤凰。
哎呀，还不知道呢，马上就知道了。明年我们明年的预算到现在也没出来。
明年不是马上这12月一过，1月就开始了呀？
我们10月份以往都是十月份就知道了，现在因为
嗯，
这个大选的事儿搞了那么长时间，所以到目前为止还不知道这个新的一年会怎么样。这个新老板上台还不知道
怎么样？
会烧几把火呢。
但关税都已经提高了，不得好好支持一下科研吗？我的天，
哎呦，你觉得关税能加到做科研上来吗？真是，你怎么你好乐观的。
换个角度，总有乐观的角度。
我觉得别不用加到科研上来，给我涨点工资也行啊。
哎，要是可以 lay off 人，就可以涨工资，对吧？减了人才能涨
你你说是简单 tech 吗？
哪儿都可以。就是你看左边和右边的同事都没了，你就可以涨工资。
哎呦，你这怎么想法这么恶毒啊？这么邪恶的想法，
我感觉你们这现在的这个目标不就是这个吗？我的理解，我粗浅的理解。
这么反动的话，我们就不在公开的平台上再讨论了吧？回头我要回去被约喝茶了，再这么说下去。我有种不祥的预感，说不定这个 FBI 就在混在我们的观众当中。
我哎，
反正马上就知道了。这个不是下个礼拜，就是圣诞节之前，反正圣诞之前我们肯定知道
哇，总得让你们把节过完吧？
啊。啊，节肯
总得让你
定过完。
对啊，把圣诞节过完再宣布比较好吧？
那看呗，看吧，看吧。
嗯，
咱们差不多开始好不好
成。
？OK，好，等一下啊。
Recording in
Okay,
progress.
so let's get started. So um, hello everyone. Welcome to our today's Museum seminar. So it's really my pleasure to welcome Dr. Zheng Wenwen from Tsinghua University to share her latest work with us today. So Wenwen actually currently is a full professor in Institute for Immunology and School of Basic Medical Sciences in Tsinghua. So, so Vaman actually has already been invited to our platform and then talk about her science a while ago. And then I think she actually really did a remarkable progress on her research program. And that's why I think it's worthwhile for everyone to learn her latest science. So very brief introduction about Wenwen. So Wenwen completed her undergraduate from Tsinghua and then continue her PhD training in genetics and development in medical science in UT Southwestern Medical Center with James Chen. After that, she actually did a quite challenging postdoc training, first in Genentech in Department of Immunology, and then she sort of redirect her research interest and relocate to Rockefeller to work with Lab of Molecular Genetics with Jeffrey who's working on neuroscience and then metabolism. So from 2014, she came back to China, started her research program in Tsinghua, and all the way up to full professor last year, as just mentioned, full tenure professor in immunology, Institute for Immunology. So one's lab actually mainly focus on physiological and pathological machinery, which underlying brain-body connection with an emphasis on the regulation function and dysfunction of sympathetic and then sensory nervous. Her lab explored the critical roles of immune and nervous systems in metabolic process, aiming to advance neuroimmune-based strategy for metabolic reprogramming. At the moment, her work actually really dive into the machinery of activation and the suppression of the host innate immune defenses and driving the development of novel therapeutics and protective strategies targeting infection and then inflammation through the host immunity. Most of the work in Wenwen's lab actually largely contribute to our understanding of the neuronal regulatory mechanisms, which can governing the metabolism and then expand insight into the neural underpinnings of the pathological conditions such as obesity, diabetes, infections, and neuropathogenic pain. And then also inspire innovative approaches for developing new therapies for the neuronal activity modulation. So as I just mentioned, women's work is really disciplinary and then across different fields to connect the immune system and neuron system. Um, Her work has been earned a lot of awards, which including but not limited outstanding young scientist funds, excellent young scientist funds of the National Natural Science Foundation in China. And also, she won the Outstanding Young Scientist Award for the Qiu Shi Foundation Career Award for Women Scientists from Chinese Society for Cell Biology. So, well, I'm a big fan of Wenwen's work. So, I'm very much looking forward to a talk. Please go ahead. Thank you for coming to our platform.
Thank you. I want to thank Chuan for the extremely nice introduction. It has been really, really supportive to not just to me and all the community. I think we are all very grateful to have this platform to interchange our ideas on science and also potentially building collaborations based on this communication. So I'm extremely grateful. So today I'll mostly share our research on the sympathetic regulation of metabolic activities. So I'll just briefly overview the sympathetic response and also sympathetic nervous system. So sympathetic response has been largely known as a fight or flight response to stimuli, particular stressors. For instance, in response to life-threatening conditions, the sympathetic nervous system can be quickly engaged. That causes the activation of adrenergic receptors within many effector organs and also signal to the adrenal medulla in the adrenal gland. So that can occur in the time scale of seconds to minutes and mobilize the body to defend the host against the many foreign threats. And in the time scale of hours, we'll see activation of hypothalamic to pituitary axis, for example, including production of ACTH, TSH, JH, FSH, or LH. That will signal to many of the organs to engage the endocrine system. Well, in days to weeks, if that stimulation persists, we'll see alteration, for example, in the inflammation, immune system, alteration in cytokines, mediators, behavior. So that will cause a lot of consequences. So that's really part of important aspect of physiology as well as pathophysiological aspects. So in terms of sympathetic nervous system, we know for a long time that that's stand for the fight or flight response. And it's known most of the cases it counteracts with parasympathetic nervous system. So for example, in many of the organs or tissues, we would see the innervation of sympathetic nerves that can, for example, dilate pupil, inhibit saliva production, dilate the bronch in the lung, and also accelerate heartbeat, stimulate epinephrine or norepinephrine release from the adrenal gland, and stimulate glucose production from the liver, inhibit the GI tract, for example, stomach, and also as well as pancreas and intestine functions, and also inhibit many other functions as well. So one of the key function it can be involved is the metabolism. So it can regulate lipid and glucose metabolism. It can alter immunity, as we have seen with many of the recent studies. It can also, of course, cause alteration in the behavior and the emotion. So on the signal transmission level, we know the sympathetic nervous system is mostly descending from the central nervous system, and the neurons receive the stimulation from the brain and then use the preganglionic fiber to extend to the ganglia in the peripheral, and then those ganglia extend the postganglionic fibers into effector organs, including many of the organs we are familiar with, for example, liver, gut, and others. Also, the sympathetic nervous system can extend to the adrenal medulla, and the adrenal medulla can largely produce epinephrine, also called adrenaline, to be secreted into the circulation and then work on the effector organs as well. So this is on the molecular level how it's transmit the signal from the central to the peripheral organs. So on the other side, we know the metabolic capacity can be quite plastic because many of the metabolic organs have to deal with the energy storage and energy demand requirements from the body. And taking the adipose tissue as an example, the adipose tissues are really heterogeneous across different spots. For example, in rodents, as well as the recent evidence show in humans, we have thermogenic adipose tissue, including the brown adipose tissue and the beige adipose tissue. And we also have white adipose tissue to take in charge of the lipid storage. And so in terms of metabolic activities, we know the brown and the beige are in charge of energy expenditure and thermogenesis, while white adipocytes are in charge of the energy storage, leptin production to maintain the energy homeostasis. So based on the early studies, we know the brown adipose tissue, which largely responsible for producing heat, are highly activated by the sympathetic nervous system. So basically, the brain can control the brown adipose tissue quite tightly because of the sympathetic nervous system. It can release norepinephrine, the neurotransmitter, to the tissue, quickly mobilize the thermogenic ability, then produce heat to maintain body temperature, especially important for rodents. was known for a long time how the white adipose tissue can receive the input from the brain, because it was largely known that the white adipose tissue are for energy storage. So the original assumption was it doesn't need the direct innervation from the brain. So a few years ago, we started our study by showing that actually in the white adipose tissue, it was not the case as we assumed at the beginning. Instead of like what we presumed, it might just receive sparse innervation from the sympathetic nervous system using, for example, synapsophysin PDP 9.5 or tyrosine hydroxylase TH. We actually see in this Y pad, we saw really dense innervation of the sympathetic nervous system. And also by quantifying many of the cells, for example, tyrosine hydroxylase labeled nerves and the paralipin labeled white adipocytes, we can see almost more than 90% of the adipocytes are possibly receiving direct input from the sympathetic nervous system. So that led us to speculate that those nerve fibers may play really profound and precise roles on regulating various cell types in the tissue. For instance, one of the cell type we got into at the beginning was the beige adipocytes in the tissue. Those we can see the UCP1 are producing cells. They are largely formed after 7 days of for example, post-cold exposure, and they are very colocalized with the nerve fibers. And we can see the nerve fibers are really in proximity to the lipid droplet filled adipocytes. So basically, we can see the synaptic vesicles along the fibers. They can be released whenever the neurons are fired. So in addition, when looking into the human Y-fat, we saw that in the human Y-fat tissue, for example, the epiploic, pelvic, mental, or abdominal wall from the human patients, we can use TUJ1 to label the total nerves or TH to label the sympathetic nerves. We can see they are largely overlapping. So basically, the human adipose tissue also receives sympathetic innervation within the tissue. And also to look into how those nerve fibers are regulated, we noticed that some of the tissue residential cells actually produce nerve growth factors. Particularly, this study we found the stroma cells produce a large amount of nerve growth factor called NGF. So we use PRX1 to drive deletion of NGF in the tissue. We can see that after NGF deletion, we really ablate the nerve innervation into the tissue. We use PDP 9.5, we can detect the loss of innervation in the tissue. And if we use tyrosine hydroxylase TH to label the nerves, we really, the innervation into the white adipose tissue was lost. So basically, the stroma cells determine the innervation establishment in the tissue, and this actually happened very early in the development. And if we put those animals in cold environment, we actually see those fat pad has defects to generate heat-producing cells. So basically, they have defects for thermogenesis. And so by doing the adipose tissue, both in the mice and in the human, we conclude that basically those white adipose tissue receive dense innervation from the central nervous system. And upon cold exposure, they actually undergo dynamic changes. They increase their density to adapt to the energy demand. And on the other hand, if we put the animals, for example, using the high-fat diet-induced obese and type 2 diabetes model, we can see actually the nerve degenerates. So basically that caused the animal to enter a vicious cycle. The brain lost control of the white fat, and that's also lead to deterioration of the metabolic states in the adipose tissue. So basically, we show that an anatomy, function, plasticity, and establishment of the sympathetic neurons in the white adipose tissue. And of course, in the white adipose tissue, not the sympathetic innervation, we know it has many cell types in the tissue. For instance, it's highly vascularized. If we look at the capillary using GPI HBP1 or using the CD35 to label all the vascular network, we can say it's really highly vascularized. So we also show that really the sympathetic innervation actually can promote angiogenesis in the tissue. that will facilitate the energy exchange between the tissue and the rest of the body. So basically, the white adipose tissue is innervated by sympathetic nerves, has a lot of blood vessels, which has crosstalk with the sympathetic nerves. And the white adipose tissue is also populated by many cell types. Stromal cells, as I showed earlier, it can produce NGF to support nerve growth. It can also actually crosstalk with adipocytes and sympathetic nerves as well. So that study started when we look into how the white adipose tissue responds to multiple stimuli in the tissue. So basically, we know for a long time that the white adipocytes express many of the receptors that mediate the response to the stimuli from extracellular stimulation. For instance, norepinephrine represent the stimuli from the sympathetic nervous system, which function through the beta-3 adrenergic receptor to cause the thermogenic process. While the white adipocytes can receive many stimulation, for example, some of the cytokines or growth factors or many other secreted factors from liver or many others. It can also, for example, signal to thyroid hormone or VEGF, many others as well. So we started by looking into what are the predominant receptors which can be expressed in the adipocytes. So one which paid attention is called OGFR because it's really highly expressed in adipocytes. So based on the single cell study, we can see it may be even higher than the beta-3 adrenergic receptor called ADRB3 here. ADRB3 respond to norepinephrine to mediate the response to the norepinephrine from the sympathetic nervous system. So if we look at the expression level of OGFR among the tissue type, for example, epididymal, inguinal, or brown adipose tissue, we can see indeed it's highly expressed. It can be also easily detected in the human adipocytes as well. And also OGFR actually stands for opioid growth factor receptor. It's an endogenous receptor for the opioid. And if we detect, if we assess other receptors for the opioid, we actually didn't see their expression in the adipocytes. So basically, it's quite specifically expressed in adipocytes. So we did straightforward experiments by deleting OGFR in adipocytes using the adiponectin crete drive the OGFR deletion. So we can achieve deletion of OGFR in those various adipose tissue. While at either at thermal neutral or room temperature, we didn't see much difference on the body weight or fat mass. So only maybe decrease on the epididymal fat tissue. So when we put into the animals into various parameter like apparatus to measure their difference, we actually noticed one difference, which is the reduced oxygen consumption during the dark phase. Basically, that means they actually has defects in thermogenesis that falls into what we have been focusing on as well. So we found those animals actually, they consume less oxygen, they produce less heat in the dark phase when they are physically active. So this is actually due to their impaired thermogenesis. For example, if we look at their thermogenic fat in the white pad, we really, and when the animals are put in the cold temperature, they really have reduced the beta cell percentage. They also have reduced the level of those genes which are involved in thermogenic capacity, like DIO2, UCP1, and reduce the expression level, the protein level of UCP1 as well. So they really are not that competent to produce heat. And also, if we put the animals in thermoneutral condition, then challenge the animals with cold environment, they really have a difficulty to deal with the cold stimuli. So they drop their core body temperature much faster. Some of them can even, they couldn't survive in the cold temperature. So they consume less oxygen. They have reduced the level of DIO2, UCP1 levels in the cold environment. So basically, we found the defects is due to lipid utilization. So if we put the animals in the cold temperature, we found with time going on, they actually have higher level of free fat acid in the serum. So basically, they have defect to consume fat acid. If we stimulate the adipocytes in vitro by giving them norepinephrine to mimic the sympathetic stimulation, we found there that the lipid droplets are reduced. The size was reduced at a slower pace. So basically, they have a difficulty to utilize lipids. So however, if we challenge the animals with glucose tolerance tests, for example, we give them IPGT, they actually has better potency to consume glucose. So basically, they have improved glucose tolerance. And if we foster the animals to force them to utilize the energy store, they also have reduced the blood glucose level, either thermoneutral or room temperature. So basically, those animals, they couldn't use a lipid that well, but they tend to use glucose when they are facing like a cold or starvation. And at the cellular level, we also checked the respiration or glycolysis of those cells. We can see that those cells indeed they have reduced either basal respiration or maximal respiration. So basically, their mitochondrial functions are reduced, but their glycolysis is enhanced to a certain level. So basically, those cells really they screwed to use more glucose instead of lipids. So those animals, if we mimic the sympathetic stimulation by giving them norepinephrine and measure the oxygen consumption acutely, those animals really, they actually consume less oxygen, they produce less heat. So basically, in response to sympathetic innervation, we think they tend to use glucose instead of lipid for the energy supply. And those animals, though they have improved glucose tolerance during steady state. However, if we put those animals on metabolic stress, for example, giving them high-fat diet, with time going on, they actually tend to develop obesity and type 2 diabetes more easily. So basically, they are more prone to develop those disease conditions. So that now, if we challenge the animals with high-fat diet, they tend to accumulate more fat. So they cannot burn the fat that efficiently anymore. So they have more fat in the adipose tissue also. And also, if we check the glucose tolerance now, after challenging the animals with high-fat diet for several weeks, we can see that now they have actually impaired glucose tolerance and they have impaired insulin sensitivity. So basically, those animals, although they are behaving better in glucose metabolism at the beginning, with the metabolic stress going on, they actually tend to develop the diabetes and type 2 diabetes the more easily. So regarding the molecular mechanism, I don't think we know much. That's so far we can see. So by doing a lot of like co-IP followed by mass spec experiments and localization studies, the only thing we can say is might be correlated with mitochondrial beta oxidation process. For example, partially in the HeLa cells, we can see the OGFR is partially localized to MTP alpha. This is a mitochondrial localized complex subunit. And the comparison we can make is the MTP alpha is involved in the fatty acid oxidation process. So in terms of the fatty acid oxidation, we know the long-chain fatty acid beta oxidation process, it converted into fatty acyl-CoA, then transported into the mitochondrial matrix, and within the mitochondria, it undergo the beta oxidation process, and it's got broken down by the MTP complex from the long-chain fatty acid to the acetyl-CoA. So basically, this is a long process for the fatty acid to be burned. And there are like a really rare disease conditions called MTP deficiency. In those patients, they have difficulty to convert long-chain fatty acid to energy. So that is a disorder of fatty acid oxidation. So those patients, they actually tend to have really low blood glucose level, for example, after fasting. And they have acetylcarnitine accumulation in blood because those are the substrate which cannot undergo the beta oxidation. So they have substrate accumulation that was spilled into the circulation. So those two parameters, those are what we actually observed with those animals. So they really cannot undergo fatty acid oxidation effectively. They have lower blood glucose level, and we actually did the metabolites measurement. We know that in circulation, they have higher acid carnitine accumulation in blood. So they have the parameters which fit into this criteria. So that's for the OGFR metabolism function. So we know in response to norepinephrine, the adipocytes have to engage pathways to burn fat. So basically, OGFR is among the factors which promote the beta oxidation to facilitate the lipid utilization and facilitate the heat production in this process. And the ligand was, by other studies, they show is met in enkephalins produced by stroma cells. The stroma cells can be one source which produce the ligand and signal to OGFR in adipocytes. So in terms of sympathetic innervation, in the organs, we know not just adipose tissue, many organs receive inputs. For example, adipose tissue receives sympathetic innervation in addition to somatosensory innervation. And the liver also receives sympathetic, parasympathetic, and vagal nerve innervation. And the gut also receives sympathetic, parasympathetic, and also send back somatosensory and vagal sensory back to the brain. And the pancreas also receives parasympathetic, sympathetic, and vagal somatosensory back to the brain. So it's really a complex multi-way communication between the brain and the peripheral. So the next one I will talk about is the study we have done between the brain and the intestine. So basically, the straightforward experiment we did early was we checked the adrenergic receptors in the gut. So the mouse has 9 adrenergic receptors. Among those 9 adrenergic receptors, we found that the gut actually expressed ADR-A2A in different segments of the gut, like from small intestine to colon. We can see the ADR-A2A expression in the tissue. And we also confirmed that indeed it's the epithelial cells which are labeled by APCAM, which are highly expressing the ADR-A2A in the tissue. And then we can recapitulate the expression of the ADR-A2A, for example, using organelle culture as well. So basically, ADR-A2A is one predominant receptor expressed in the intestinal epithelial cells. And also in the human, based on the human single cell study, we know the ADR-A2A was also highly expressed. Although the human cells also tend to express ADR-B2, which might represent additional layer of sympathetic regulation on in the intestinal tissue. So anyhow, we made a conditional knockout mice by crossing the ADR-A2A with william-crate to delete ADR-A2A in the intestinal epithelial cells. Now we actually didn't see either IR, oxygen, or heat production in those animals at a steady state. So basically, they didn't show really dramatic defects at a steady state. Also, with time going on, like we didn't see much difference on the body weight for the intake at the scale of acute or food intake with a long stage. We also didn't see difference either like fat mass weight or fat mass percentage or blood glucose level at basal state or liver glycogen. We didn't see much difference either. So the difference, the significant difference we saw was the blood glucose tolerance test. We did the oral glucose tolerance test. We actually noticed those animals, they have improved blood glucose tolerance. So basically, those animals can process glucose much more efficiently. And when we do the IPGTT, basically we inject glucose into the gut, into the peritoneal, we didn't see difference. So that means the glucose has to pass through the lumen to be sensed and then engage the sympathetic regulation to signal to the epithelial cells. At that stage, the ADR-A2A signal is important. And also this glucose tolerance improvement we can see in across many conditions. For example, high carbohydrates condition, high fat condition with 40% of fat and 60% of fat. So we can all see, even with different metabolic stress, this glucose tolerance can be better in the conditional knockout animals in all those conditions. So that with that, we actually turn to the cell types of enteroendocrine cells, because those are the sensory cells in the gut, as many of us, and also Tran has studied that as well. And those cells actually sense the presence of many gut lumen contents, for example, glucose, among many others, that can produce many of the secreted molecules. And the GLP-1 is among one of the key hormones produced by L cells. And GLP-1 has been the drug blockbuster in the past few years because of its role to control blood glucose and reduce body weight. So that's obviously of our choice at the moment, because it's really important for blood control. So when we look at how the sympathetic nerves can regulate L cells, the first thing we look into is their proximity. So by staining TH to label those nerves, or PDP-9.5 or TUT1 to label the TUT nerves, we can all see the GLP-1 producing cells. In this case, we label the precursor for GLP-1. We can see certainly many of them are very close to each other. And both like in colon and ileum, we can all see the GLP-1 producing cells are close to those nerve types. And also across the whole mouse intestine, we can see the TH label nerves, or PDP-9.5 label TUT nerves. They are quite close to the GLP-1 producing cells. So basically, by quantification, more than half of the GLP-1 producing cells may be in a few micrometer distance from the TH positive nerves. And also in the human intestine, we can see similar cases. For example, GLP-1 producing cells, when we co-label them with THPDP-9.5 or TUT1, we can see they're quite close to each other, either in small intestine or colon. And also across the whole fragment of the human intestine, we can see TH or PDP-9.5 labeled nerve fibers, or GLP-1 labeled L cells. They can be very close to each other. So even in human gut, we can see in like a 2 micrometer range, we can see more than half of the L cells are actually located within certain nerve fibers. And so when we look at how the sympathetic innervation might affect those L cells, we didn't see much difference on like expression level or total level of GLP-1 precursor in the tissues. But if we look at the GLP-1 production, it's actually a tight regulated process, a signal dependence in presence of luminal glucose. It engage the signaling cascade to engage the GLP-1 release into the tissue. So if we look at the releasing process, we notice that in response to glucose challenge, we saw the GLP-1 actually, we saw less GLP-1 left in the tissue. So basically, that indicates the GLP-1 was released more potently in those conditional cow mice. So that actually means the model we proposed. So ADR2A signal is a negative regulator of GLP-1 secretion. So when we release the ADR2A signal, we actually have more GLP-1 release. Therefore, we can detect less GLP-1 in the tissue. So basically, it's a negative regulator of GLP-1 production. So when we looked up to see whether the signal from the sympathetic innervation can affect the GLP-1 release, we crossed the TH-CreART2 animals with HM3DQ to use the chemogenetic activation of sympathetic nerves in the animals. So basically, in this case, if we give the animals CNO to activate the sympathetic nerves and then challenge the animals with glucose, now we have more GLP-1 left in the tissue. And we also tried the viral-mediated sympathetic activation. So basically, we did intracolonic injection of retrograde AAV virus. We injected the TH-CRE into the HM3DQ animals to drive the HM3DQ expression in the sympathetic neurons, which projected to the gut. Now, if we looked into how the virus can label the sympathetic ganglia, we saw we have a significant level level of the viral tracing into the ganglia. We saw citrin expression in the ganglia. We can zoom out some of the neurons which are labeled by the retrograde virus. And in those animals, if we activate the sympathetic neurons with CNO, we can indeed see the glucose tolerance was impaired. Basically, when we activate the sympathetic neurons, we have worse glucose tolerance. So now, if we look at the GLP-1 in the gut, we saw that when we activate the sympathetic neurons which protect the gut, we actually have more GLP-1 left in the tissue. So those are chemogenetic activation. If we use genetic deletion now, if we delete norepinephrine production by driving the DBH deletion using THC-ERT2 animals, we can get rid of norepinephrine production in the sympathetic nervous system. Now we can have the animals to be challenged by the glucose. And if you look at the GLP-1 in the tissue, we actually saw less GLP-1 in the tissue. So basically, if we release the sympathetic suppression, we saw more GLP-1 production and secretion from the L cells. And in those animals, if we look at the insulin sensitivity, we actually saw though they can preserve the insulin sensitivity for a long time. That is different from the drug administered situation, because in the GLP-1 drug-treated humans, the theory was GLP-1 actually increased the production of insulin from pancreas. Therefore, it can control the glucose level. But in our case, in the endogenous GLP-1, it doesn't seem to be the case. It looks like those animals actually have higher sensitivity to insulin, and in response to glucose, they don't need to produce that much of insulin. They actually have less production of insulin. So that means the endogenous GLP-1 possibly function through a insulin-independent manner to control the blood glucose level. And also the insulin sensitivity can be preserved even like in the metabolic stressed animals. For example, with high-fat, diet-fat animals, we can still see the insulin sensitivity that is improved in the conditional knockout animals. So for the ADRA2A signal, we actually have been curious if the ADRA2A signal in the L cells is actually not a good sign for the metabolic health, why the animals still keep the signal for a long time during evolution. So therefore, we actually mimic the natural situation by giving the animals limited time to access to the calorie. For example, we only give the animals for 1 hour every day, so they can only take 40% of the calorie into the during that time period of time. So now if we check the body weight and the blood glucose, we actually saw those animals really have a difficulty to keep up their body weight. So basically they have a difficulty to maintain their blood glucose level. That will cause problems if they have to deal with the threats. So this is how we reason the conservation of this signaling axis. So basically on the molecular level, we also, with the data I'm not showing here, we also show that the ADRA signal possibly function through as a GPCR-GI couple signaling and inhibit the level of cyclin AMP. Therefore, it can decrease the secretion of GLP-1 from the L cells. So the last question, actually, we asked if the glucose was reduced in the blood, where it actually go. Because we tested the absorption of the glucose into the system, we think that the absorption might not be the significant contributor to the reduced glucose in the circulation. So we looked into where the glucose can go by using the relabeled glucose scanning by PET-CT. So one region we noticed is the brain, because in the CQ animals, we found that it has faster and more glucose uptake into the brain over time. So we think one possible region the glucose can go from the circulation is the brain regions, actually. So we confirmed the glucose utilization in those brain regions. For example, one region we tested is the hippocampus. So we compare the glycolysis process in hippocampus by comparing the wild type and sickle animals post glucose uptake. So we noticed that those animals actually tend to have a higher level of the the intermediates in the glycolysis process. So basically, those animals seem to utilize glucose more, possibly due to the increased uptake into those regions. And the consequence of this, we can see that from behavior tests. For example, if we check how the glucose utilization in the hippocampus can affect the animal, we put those animals in the Morris Water maze to determine their the cognitive function. So basically, we noticed that with time going on, those animals actually spend less distance to platform. They have reduced the latency. They spend more time in the target quadrome. So basically, they can quickly find their targets. One reason being that was they utilize the glucose more efficiently in the brain regions. And so because we started from GLP-1, those animals tend to have more glucose utilization in the brain. So we touched upon little how the GLP-1 signal might affect the glucose utilization in the brain. So one possible contributor is the glucose partition into the brain. So we noticed that when we give the animals exciting food to engage the GLP-1 signal, we actually saw the enhanced oxygen saturation in the brain circulation. So we measure the oxygen saturation we saw in the CQ animals. We actually have slightly increased oxygen saturation. So basically, they seem to exchange more of the energy or substrate with the peripheral. And also in terms of sterile vasodilation, we also try to quantify the vessel diameters. We saw slightly increased vessel diameters right after we give the animals exceeding full. So basically, that might contribute to infused glucose from the peripheral into the brain regions to supply the brain energy demand. So in this part, I think we found the sympathetic nerve endocrine unit in glucose control. So when we have high sympathetic activity, the gut GLP-1 production can be reduced. That will cause problems in the blood glucose level. So that have increased blood glucose level and reduce the brain glucose uptake. That actually will affect the cognitive abilities. For example, of course, on the counterpart, there are like vagal nerves and other neurons which are involved to counteract the glucose metabolism. So overall, we think the sympathetic activity decreases GLP-1 release and reduce the glucose tolerance. And ADRA2A signal on L cells seems to be a signal pathway which limits the GLP-1 secretion. And the sympathetic L cell unit can reduce glucose utilization in the brain. And this utilization reduction in the brain can hampers the cognitive function. So as a summary of my talk today, so basically I show that the sympathetic nervous system really function as a key bridge between the brain and the peripheral. So basically either for the adipose tissue or for the intestine, it can regulate key functions of those tissue for like thermogenesis, for endocrine functions, for glucose control. And also if we look at more comprehensively of the sympathetic nervous system versus the parasympathetic nervous system, we know the sympathetic nervous system can be involved in many more functions we don't understand yet beyond what we currently understanding. For example, in addition to metabolic organs, we know it can regulate like immune organs and reproductive organs and respiratory system, many of them. So I think we still have long way to go to fully understand how the signals can be integrated to really maintain the metabolic health as well as the overall survival benefit to the animals. So with that, I'll stop here. So I would like to thank the students who have done the hard work, our collaborators, and also our founders. So thank you, and I'm happy to take questions and also interact in the future. Thanks.
Recording stopped.
Well, as always, Wenwen's data is really a lot. uh,
Work hard.
yes, work hard. So we can start a question section. Anyone have a question, please just either raise your hand or put your question in the chat box. Xiaofei, go ahead.
All right. Okay, let me see. Hi everyone.
Hey.
Good to see you again. Hi there. Yeah, it's a very beautiful two stories, and actually I have like two one question related. The first question, like when you see like when you active the sorry, when you suppress the sympathetic nerve, you increase GLP-1 production, and under that situation, so you see like increase insulin sensitivity in these animals, right?
Mhm.
So under that situation, have you checked if that's actually directly because of GLP-1, or it's because like innovation of, for example, muscle tissues or or liver tissue?
That's a very good question. We actually don't know because there's certainly direct innovation to all those organs. It's possible it can be both. So the only thing we can say is it's systemic. It's possibly not due to GLP-1 alone.
Mm-hmm.
Yeah.
Yeah. So I think like in one of our studies, we actually try to increase gut GLP-1 production using using a bismuth drug, and under that situation, we also see increase in insulin sensitivity. So I guess like kind of point to the direction you are trying to point to is that probably GLP-1 has some function on insulin sensitivity itself. But anyway, I feel like maybe other neurons can also participate in this process.
Yeah, it can be direct or indirect. So direct maybe because the genetic knockout we have been using is systemic genetic knockout. It can be direct to the pancreatic. But like you said, the insulin sensitivity can be directly altered maybe by the GLP-1 signaling as well, because the signaling pathways themselves can be crossed out, but those cells can express the GLP-1 receptor. So that can be one of the contributors as well. But the other the confronting factor may be the lower glucose level, because you keep have lower glucose level, that might lead to change in those insulin sensitivity as well. So I think those all three factors can, I don't know, maybe you can like clear that up. Maybe the which one is the dominant factors which contribute to the improved insulin sensitivity. But so far we can speculate as all those three factors can all contribute to that part. Yeah.
Okay. My second question is that related to your last working model. And in that model, you said during sympathetic activation, you have reduced brain glucose utilization, and okay, so it's kind of like counterintuitive to me, obviously because I don't know much about that. So like I know like when animals during like under stress conditions, they're gonna increase some glucose mobilization and to fight or flight. Either way, you're gonna need a lot of glucose. So I feel like under that situation, do you need your brain to be like, really ready to make those decisions and like work really hard on that moment? Why do you kind of like, have to reduce glucose utilization for the brain?
That's a good point. So for the sympathetic activation, we need to actually check whether systemically the sympathetic activation can indeed cause the reduction in the blood into the brain. So that experiment was done particularly using just GLP-1 signal stimulation. But we don't, because the sympathetic activation actually extends to many of the organs, we don't know the overall consequence of that. But the thing so on the physiological level, what we know is in terms of the fight and flight, the blood circulation into the muscle and the heart can be increased, because those two tissues are really the ones you really need the most. Like you need the heartbeat. Yeah, increase, you need the muscle to fight. Actually, for the brain, we really need to look at that carefully. Even, for example, different regions brain, they might be actually need different, they have different energy demands. So overall, I don't think we know the exact answer for that. Yeah.
Okay.
But that's an interesting point. Yes.
I feel like maybe like under probably very unlikely situations, you have energy competition between, for example, brain and the peripheral during those like stressful conditions.
Yeah, that's true. Yeah, that's true. Because during that like really short time period, the energy allocation can be really critical for the animal. That's the reason it really has to be engaged in really fast to deal with that. Yeah. Yeah, I
Yeah.
agree with
Okay.
that. That's a good point.
All right.
Yeah.
Cool. Thank you very much.
Thanks.
Hey, well, I think I'm gonna start also questions that the expression pattern of you show the ADRA2A seems like highly expressed in the colon compared with small intestine. And then I mean, but I'm actually curious that why the because I know that the absorption of the glucose or the utilization of these usually happen beyond the colon part,
Mm.
and how you reconcile this phenotype.
Yeah, that's a good point. Actually, so at the beginning we thought the signal can be really general to the enterocyte. That's the reason we use Villian Cree to drive the deletion. So, but as you must be very familiar with, actually enterocytes are really complex composition, has many different cell types, and even on different location they can have different functions. So we actually didn't try to sort that out. We tried to use different Cree, but those Crees are not really specific to certain cell type. For example, GLG Cree and some enteroendocrine cell Cree to address that. But in terms of enterocytes, we know the innervations are not homogeneous. The receptor expression are not homogeneous. And even there are some other receptors which are expressed different in different cell, they can be vary as well. So that might not be like solely attributed to the control of glucose. So glucose may be one of the aspect it actually contribute to, but in terms of the colon and function in the colon, it might be have many other aspects. It also regulate. I don't think our results actually have been able to sort that out. Yeah.
So then the follow-up question is, although you see the expression of this ADRDA2 alpha in the epithelial cells, I mean, but you did look into the single cell. Do you know how the expression pattern on the epithelial cells?
You mean among epithelial cells?
Yes, among the epithelial cells.
They all express, but I don't remember like a relative level whether like we only looked at L cells and we made sure they are expressing that. We didn't look at like variation among different like their relative quantities. But that's a good point. They actually could make difference. So the relative level of different receptors can make difference. Yeah,
Yeah, right.
but yeah.
That's what I mean. So I mean the phenotype, obviously the total Cree cannot cannot determine that the L cells are the dominant one which contribute to the
Right.
yeah.
Yeah, it's so the only thing we can say is so the stimulation probably mostly engage the L cell. But if you look at other cells, they also express this receptor. And under which scenario they might be involved, I think that's different story. It's possible under different scenario they are engaged too. Yeah.
Right.
Yeah.
There's a question from the audience, Eugenio. Congrats on the work. The question is some bacterial, I think, produce metabolites that activate ADRA2 alpha. Can the activation of this receptor from the microbiome metabolism to associate with cognitive impairment? And then the increased GLP-1 can also change the microbiome. Have you considered evaluating the microbiome in the Villain-Cree, your conditional knockout mice?
That's an interesting point. I actually believe those cells must be like really in a key position to mediate the crosstalk between the gut microbiota and the sympathetic nervous system and even like enteric nervous system, all of those. I think it's certainly be one of the key points we should consider when we consider how the microbial metabolism or how the microbiota can talk to the host. That can also change how the sympathetic nervous system may be involved. But I don't know the exact answer for that.
But have you checked the microbiome on the
Yeah,
mice?
actually, the first thing we checked was microbiota.
And
We saw some difference, but we didn't understand the contribution of that difference linking to glucose level. We actually seeked the microbiomes, the S and all the microbe, the sequencing. We done the meta sequencing at the beginning. Yeah.
But let's say if you like say put the mouse together, co-house them, can you somehow change the phenotype?
That's a good point. We didn't do that.
Okay. Okay. So
we
there,
saw we noticed some difference, but since we cannot do we don't have the like expertise to do like perturbation study, loss of function controls, do all kinds of those stuff. So we stop there. So we only keep that in
yeah,
mind.
should ask me.
Yeah, maybe I'll pull out the data and let you take a look.
My next question is, you say that obviously the knockout have actually changed the GLP-1, which being compensatory utilized from the brain. But my question is, it seems to me that the GLP-1 difference will also change the vasculature. I don't know about the density or the diameter, but it changed the structure of the brain vessels. But why how that happen?
Structure.
I mean, you say there's increased utilization of glucose in the brain due
Mm.
to the vasculature. I don't know if it's structure or it's the diameters of the vessel.
It seems, that's a good question. See, actually, it's the response to the stimulation, rapid response. It's the diameter change acutely. So
then my
we
question
keep on, yeah.
my question is why?
So one possibility is GLP-1 actually can engage the ascending neuron pathway. For example, we go neurons to the central nervous system. That can actually cause the descending signal to alter how the brain interprets those signals. So exactly, I have to see, I don't know the exact answer. That's purely my speculation. So there are some studies showing the astrocyte vesicular glial coupling can be altered actually by GLP-1 signal. So the glial vesicular coupling in the brain, when the coupling is altered by the GLP-1 signal, that can actually change the circulation into the brain. But the signaling pathway can be direct stimulation on the neurons in the brain regions. For example, some of the neurons which can certainly express the GLP-1 receptor, and that can also caused by the afferent nervous system. For example, you have the glucose when you have the vagal neuron engaged, that can actually send the signaling to multiple brain regions, and that the brain activity change can be coupled to the vasculature, the blood absorption into the brain regions. Basically the neural activity is coupling to the neuronal activity.
Well, I
Either
mean,
direct or indirect.
well,
So direct,
no, go ahead, go ahead.
so the direct is through the direct activation of GLP-1 on GLP-1 receptor on neurons. So once the neuronal activities are changed, they change the glial vesicular coupling. So that will cause the blood circulation alteration in the region. The other one is the ascending neurons. So we know the vagal neurons certainly can respond to the GLP-1 signal. Once the vagal neurons respond to the GLP-1 signal, that can cause the ascending neuronal network, the circuitry change in the many of the brain regions indirectly. So that will cause the neuronal activation in all those regions and causing glial vesicular coupling alteration as well.
But this all these things you describe actually should be have been validated in the GLP-1 knockout mice, right?
So there are many indications. For example, knockout animals. I don't know about the knockout animals. So that has been validated. That has been many indications for the GLP-1 receptor agonist treated situations. For example, you have improved glucose metabolism in the brain and particularly in the Alzheimer's disease patients. So the potential benefits brought by GLP-1 was considered as the glucose utilization in the brain regions. So you improve the glucose metabolism that can cause beneficial effects.
And all these beneficial effect is because specifically neuronal receptor of GLP-1 induced?
I don't think so. So that actually so the experiments we done, we have a few caveats and gaps in the experiment. So that experiment was done by using exidin-4. Exidin-4 itself actually has off tissue, although on targets, but off tissue effects. So we have to like really, if to answer that your question, we really have to do more experiments. For example, using the GLP-1 receptor.
You have done enough. You have done
no, so
too many.
that part are really loosely related. I cannot make like a really rigorous conclusion from.
I see. No, I'm actually just curious that must be have a lot of study based on the GLP-1 receptor and then related to the cognitive responses. And then I was just wondering whether these study actually all correlate with the receptor back to the neuronal sources, or it's just other sources also contribute.
Based on the literature, there are many sources which can contribute.
So,
right? But I don't know which one actually prevail. I mean, which party actually prevail?
I see. That's interesting. There's another question from Izzy. So does ADRA2 alpha deletion change glucose absorption?
We have done some experiments, at least from the more rigorous experiment. What we should do is actually in vivo. We did some in vitro experiment by just like isolating fragments to do that in vitro. We didn't see much difference on that part.
Okay. But that's.
I have a question.
Yeah, go ahead, Paul.
Hi, Wanda. Very nice talking. So interesting. So I have a specificity question. So you basically attribute the whole phenotype to L cell. So the specificity question I have is that L cell also produce GLP-2 as well. Like I understand you focus on GLP. I just want to have your comments. Can you comment about, like, for example, L cell producing GLP-2, or have you explored the K cell, which will express for example, GIP, sorry. So like, why you specifically just target GLP-1, not GLP-2 or GIP in not just only L cell but also K cells?
Yeah, that's a really interesting question. Actually, that's a question we also been asked by the reviewers too. So I think the contributor can be actually achieved from many of the factors, but depending on the stimulator you give that might engage predominantly different cell types. If we change the stimulation, we might be able to see difference on the GLP-2 as well as GIP-1. And also because as you can see, actually those staining, they are not that specifically targeting GLP-1 itself. Actually, we use a precursor type to monitor the level. So to be honest, I don't think our conclusion actually excludes the involvement of all these others. In order to exclude, we really need to specifically block the function of GLP-1 and exclude the others. I think it's possible those others also contribute to. Yeah, I don't think our experimental evidence actually excluded their involvement, to be honest.
So if it's GLP-1, first of all, have you used L cell Cree to test the hypothesis, or with this receptor knockout, you also knock it out the GLP-1 to see if your glucose postprandial glucose phenotype is still preserved?
Yeah, that's a good point. Well, actually, if given time, we should do that. So the only Cree we tried is we have been thinking about trying with the GCG Cree, but that also target to pancreas. So by far, we don't have a really specific Cree to target just L cell, but not touching other endocrine cells, either in the gut or in other organs. So I'm not sure. Maybe you have suggestions on the Cree line we can use. So that's actually the technical challenge. So when we found the phenotype of William Cree, the first thing we thought about was we need a specific Cree, but we actually spend some time. We think that will take a long time to get that. That's I agree. That's actually one of the caveats we admit. Yes.
And another way to ask the similar question, have you challenged mouse with the oil instead of the glucose to see, postprandial lipid output can be also effective or not?
Yeah, that's good point. We actually done some experiments. We give them a lipid, we measure the fat acid in the blood circulation. We did a little bit of that experiments. We actually saw, if I remember, we saw some defects on that part, but we actually saw a little bit to opposite defects. But we need to really verify that experiments. We haven't been able to like really rigorously using multiple experiments to confirm that. We saw some difference. Yeah.
Okay. And now I want to also have your input about the conceptual idea. Because as I recall, this is what I don't understand because I don't do neuron-related research. This is what I learned from, for example, Ruslan and Dan Lindman a couple of years ago. They published a paper. Both of them say that, postprandial sympathetic nerves can, affect the immune cells, including gamma delta T or ILC3 through the VIP. Regulation, it will affect the IL-22. And then later on, what the layman want to say is that, during a postprandial effect, the neuron activation, at least is a VIP nerves activation will help the lipid uptake through intestine. So in other words, the nerve activation actually, helps the lipid processing. But in your case, you're saying that, the nerve activation actually reduces the GLP-1, right? And then it actually, inhibit the glucose processing. So, like to me, I kind of a little bit confused, like during, postprandial effect. I mean, why, the nerve activation have different, effects. I mean, your mechanism different from what they want to say, to my understanding.
That's a great point. That's actually what we are trying to understand by far too, because we know the innervation can. So, for example, even to the same organ in different cell types that might function differently. For VIP, so from if I remember correctly, that's actually a signaling between the like sympathetic nervous system and the enteric nervous system together with the downstream signaling. So that can be involving other partners in between. So for example, the enteric nervous system, enterocytes, and even the stroma cells in the gut, they can, so if you look at the innervation, they are really densely distributed within the gut system. It's possible when those cell types, they respond to the innervation, they don't respond like homogeneously. They either produce some of the hormones more, they produce less of the other hormones. So they actually alter the metabolic activities, but it's not like that. You cannot simplify that as like enhanced glucose or inhibit lipid. And even for glucose, they are not. We cannot like call that like enhance or inhibit. We can only see they change the glucose partition into different organs. That can maybe a more precise description. So using the system, they might actually fine tune how the energies are absorbed or allocated, stored, distributed, those kind of stuff. So that's what we try to propose. At least I think that is in line with our study as well, because those are like a fine step of those all the processes. Yeah.
I see. Yeah. Thanks for the comments. Yeah. Very interesting.
Thanks.
Okay, so I do want to.
There's one question. Is the question PX?
Yeah, you just I think you just answered the question.
Oh, the PRX1, just the internal. Okay, I don't know whether.
Oh, yeah. There's one more question. Yes, there's a question in between. Ming Lu have the question regarding the story of PRX1 Cree NGF flux mice. Since PRX1 Cree may predominantly label the limbo, but mesenchymal cells. Have you checked the other mesenchymal cell line to address their role in the nerve, the
Okay. I
generation,
think
internal
yeah,
organ?
yeah, this is also about the specificity issue. Actually, we have all with all the crees. We know the cree certainly not perfect. It can also target other cell types. So the only thing we can say is in many other organs, we also checked whether this cree can drive their deletion. We know it's not like universally driving NGF deletion in many other organs. For example, in gut and in other organs. So we know certainly it has certain specificity, but not the specificity like she's mentioning here. So we haven't used other Cree. So PRX1 is the Cree we had back then in hand, which is the best aligning with the NGF expression in fat. Yeah. But thanks for the question.
Okay. Okay, so yeah, so there's if there's no more questions from the audience, uh, if anyone interested in Wenwen's work, just to go ahead to check her paper. There's a lot of data. I can assure you, you you will learn a lot. And if you have more questions about her work, I think I will be happy to take a question by email. If there's no further questions, then Owen, thank you so much for sharing
Thanks,
the
man.
work with us today, and then very much looking forward to your next work.
Thank you.
Yeah,
Thank you.
yeah,
Thanks, John.
thank
Thanks, Paul.
you.
Bye,
Yeah,
Paul.
yeah. Bye bye,
Looking
Bye.
everyone.
forward to
Bye,
seeing you again.
bye,
Bye.
bye.